Hello everyone, I'm Alexsandre Gutierrez-Barragan, a graduate student in biosciences at the Universidad de Sonora, and today I will present the results of the genetic characterization of the Yaqui Catfish reproductive stock of the Bavispe River sub-basin in Sonora, Mexico. Well, the Yaqui Catfish is the only described native species of Ictalurs in northwestern Mexico and the southwestern United States. Historically, Yaqui catfish have been collected in the Sonora, Yaqui, Mayo, Fuerte, and Casas Grandes river basins. However, for several years it was declared extirpated from the Sonora and Casas Grandes river basins, and Varela-Romero et al. In 2011 did not detect Yaqui catfish in the Mayo River either, and currently Yaqui catfish have been collected only from the Arroyo Cajon Bonito and the Tutuaca River in the Yaqui River Basin, and from the Baropilas and Garachi rivers in the Fuerte River Basin. Due to this loss of populations, in Mexico the species was considered as threatened by the Norma Oficial Mexicana 059. Also, the red list of the International Union for Conservation of Nature classified it as endangered. And the main threats to the Yaqui catfish are habitat alterations, over-extraction of water, among others, but the main threat has been competition and hybridization with the channel catfish, which is an exotic species that was introduced to be used in aquaculture. The first hybrids were observed in the 1980s in the Yaqui and Fuerte River basins. This was corroborated in 1990 by morphological and osteological analysis by Miller, and later corroborated with allozyme analysis in hybrids of catfishes collected from the Aros River in the Yaqui River Basin and used to create a reproductive stock in Dexter, New Mexico. Later, Baker et al. In 2008 indicated possible hybrids in Yaqui catfish populations that had been released in Arizona, although a subsequent microsatellite analysis did not detect hybrids. Due to this problem, there is a binational Yaqui catfish management and conservation plan between Mexico and the USA. In 2019, a reproductive stock was created at Rancho San Bernardino in Mexico, with individuals collected from the Arroyo Cajon Bonito in the Bavispe River sub-basin. These individuals were identified by the morphological diagnostic characteristics that I mark in this figure. However, the hybrids can present intermediate characters or a mosaic of parental phenotypes, so it is important to carry out a genetic characterization of the reproductive stock individuals to evaluate their purity. Therefore, the main goal of this work was to develop a genetic characterization of the Yaqui catfish reproductive stock from the Bavispe River sub-basin. The first goal was to evaluate the phylogenetic relationships of the Yaqui catfish reproductive stock through the site B and COX-1 mitochondrial genes and the RAG1 and RAG2 nuclear genes inconcatenated, and the second goal was to determine the specific identity of the Yaqui catfish reproductive stock founders candidates using the character-based DNA barcoding of the COX-1 mitochondrial gene. The study area is the Arroyo Cajon Bonito. Here, in 2019, 18 catfish individuals were collected, which were transported alive to a pond at Rancho San Bernardino. Each individual was marked with a pit tag, and tissue was taken from the adipose fin, and one Yaqui and one channel catfish that had been collected in 2017 in the Arroyo Cajon Bonito were used as reference samples. Also, 4 channel catfish specimens were collected in culture ponds upstream from our study site. From these tissues, DNA was extracted and the mitochondrial and nuclear genes were amplified by PCR. These genes were sequenced in macroGene Korea. The sequence editing and assembly was carried out in Chromas Pro and the alignment in Clustal W. For the phylogenetic analysis, the best fit nucleotide substitution model was obtained for each gene in J-model tests on Cypress. The maximum likelihood analysis was developed in POP, and a post-hoc bootstrap analysis was performed in RAX-ML on Cypress. And the DNA barcoding was obtained from the first 651 base pairs of the COX-1 gene. First, genetic distance were obtained in MegaX, then an ever-joining tree was obtained in PUB, and in Mesquite, the Nexus tree file was generated to obtain the character-based DNA barcoding in the cow software developed by Sarkar et al. Well, the phylogenetic trees obtained by maximum likelihood for each mitochondrial gene and inconcatenated show the same topology. Here I show you only the phylogenetic tree of the site B and COX1 inconcatenated and using Ameiurus nebulosus as an outgroup. The topology was well supported by high bootstrap values and showed the Ictalurs genus as a monophyletic clade. Here the Furcactus and the Punctatus clades were resolved, both clades defined by Lundberg. Within the Punctatus clade, the channel catfish shared a common ancestor with the Ictalurus lupus, which in turn is well differentiated from the Yaqui catfish, and this topology is the same as previous morphological and molecular phylogenies. With respect to the individuals of the reproductive stock, 15 individuals form a clade with the Yaqui catfish reference sequences from the Yaqui River Basin. However, individuals 2, 4, and 18 form a clade with the channel catfish, indicating their maternal channel catfish lineage. We did not find evidence of ancestral haplotype by incomplete lineage sorting, and no evidence of pseudogenes or heteroplasmy was found too. Thus, this represents the first evidence of hybridization for us. Then, the hybrids were identified from the nuclear genes. Here, the double peaks in the electropherograms at the diagnostic position between Yaqui and channel catfish indicated hybridization. Here I show you the diagnostic position between Yaqui and channel catfish in the RAG1 and RAG2 nuclear genes. For example, at position 594 of the RAG1 nuclear gene, there is an adenine in Yaqui catfish, but there is a cytosine in channel catfish, and thus the hybrids were those individuals that show double peaks in this position, and it is the same for the rest of the diagnostic position between Yaqui and channel catfish. In this table, the diagnostic positions that I previously showed are summarized, and it is observed that the individuals 1, 2, 4, and 7 of the reproductive stock presented double peaks at the diagnostic position between Yaqui and channel catfish, indicating their status as hybrids. Additionally, one individual collected in 2017 and morphologically identified as a channel catfish was identified as hybrid from this analysis. Of these hybrids from the reproductive stock, individuals 1 and 7 presented a Yaqui catfish mitochondrial genome, and individuals 2 and 4 presented a channel catfish mitochondrial genome, thus indicating that the crosses are occurring in both senses, that is, between females and males of Yaqui catfish and males and females of channel catfish. And the individual 18 of the reproductive stock was heterozygous at the diagnostic position between channel catfish haplotypes A and B, indicating its status as pure channel catfish or backcross. With this information, the haplotype subtraction method developed by Clark was used to extract parental haplotypes from the hybrids to perform phylogenetic analysis. As our goal was to identify pure Yaqui catfish individuals, only the channel catfish nuclear haplotype was used from the hybrid individuals. The topology obtained for each nuclear gene and inconcatenated were the same. Here I show you only the phylogenetic tree obtained by maximum likelihood of the RAG1 and RAG2 inconcatenated. The topology was the same as the phylogeny of mitochondrial genes and as phylogenies previously reported. Here, 13 individuals from the breeding stock formed a clade for the Yaqui catfish, and individuals 1, 2, 4, 7, and 18 from the reproductive stock formed a clade with the channel catfish, indicating their phylogenetic affinity. Finally, the character-based DNA barcoding from the COX1 mitochondrial gene show 44 pure attributes or autapomorphisms that are present in the Yaqui catfish but not in the channel catfish, and it showed 3 private attributes that are present in channel catfish haplotype 8, but not in the Yaqui catfish. In summary, 4 hybrids between Yaqui and channel catfish were identified, and 1 pure channel catfish or backcross was identified. However, it is not known whether these hybrids are infertile due to postzygotic barriers or genomic incompatibilities, which would cause loss of reproductive effort, or if these hybrids are fertile by backcrossing with the parental species or with other hybrids, which will lead to a genetic introgression. When this occurs, hybrid swarms are formed, and this can happen in many populations, leading to a widespread introgression in the geographic area, or even leading to a complete admixture of parental species. However, when genetic introgression occurs, it is necessary to use hundreds of molecular markers, such as SNPs, to describe the hybridization and genetic introgression dynamics. On the other hand, the Yaqui catfish population of the Arroyo Cajon Bonito is isolated by an intermittent section, so the hybrids detected here may be due to the channel catfish escapes from the culture ponds, which currently belong to Fundación Cuenca Los Ojos, who suspended the channel catfish culture. However, this same hybridization phenomenon may be occurring in the rest of the Yaqui catfish remaining populations, so it is necessary to analyze the status of this population and create reproductive stocks for each of the remaining pure populations. In addition, it is also important that the Mexican government regulates the channel catfish culture in order to avoid the loss of the few remaining Yaqui catfish populations. So, the phylogenetic data showed 13 putatively pure Yaqui catfish individuals from the Yaqui catfish breeding stock at Rancho San Bernardino in the Bavispe River sub-basin. However, the mitochondrial and nuclear DNA evidence showed 2 hybrids with Yaqui catfish maternal lineage and 2 hybrids with channel catfish maternal lineage, indicating that the crosses are occurring in both senses, and 1 pure channel catfish or backcross was detected. On the other hand, the character-based DNA barcoding showed the characteristic attributes that allow the mitochondrial differentiation among Yaqui and channel catfish. Therefore, it is important to consider these results for the reproductive stock management, discarding the individuals identified here as hybrids or channel catfish. And what we are beginning to do is to use next-generation sequencing to obtain numerous molecular markers in the genome, such as SNPs or macro-haplotypes, that allow us to develop statistical analysis to understand the hybridization patterns and genetic integration between Yaqui and channel catfish throughout the Yaqui catfish distribution, as well as determining the genetic variability and population structure of the remaining population of pure Yaqui catfish. And finally, we thank the Desert Fishes Council because this is the report of the results obtained through the Minkley Memorial Grant of 2018 and 2019. Thanks to everyone who helped us in the field collection, mainly thank Drs. Alejandro Varela-Romero and Chuck Minkley, who are coordinating this binational effort to conserve the species, and thanks to Ana Valer Clark, Jose Manuel Perez Cantú, and Fundación Cuenca Los Ojos for allowing us to carry out the field samplings and use their facilities to start their reproductive stock. And that's it. Thank you very much for your attention. I hope everyone understood me well. This was my first time presenting in English. I will improve for the following DFC meetings. See you.